Hello, everyone. I am Marine.beraud, the communication assistant of France-BioImaging, and I'm happy to be here today to share the first edition of our new webinar series, FBI Connect. Briefly, for those who don't know, France-BioImaging is a national research infrastructure in biology and health dedicated to microscopy and bioimaging. It's organized into 10 local nodes across France, as well as one transversal bioinformatics node. Our mission is to provide researchers with access to cutting-edge imaging technologies available in our core facilities, along with high-level scientific and technical support. We created FBI Connect to share the latest techniques developed within our facilities and R&D teams with the scientific community. In each session, our goal is to introduce you to an innovative microscopy-related technique and show its concrete applications so that it may support your research work. FBI Connect is also an opportunity for FBI members to discover what is being developed across infrastructure and to foster future collaborations. And for this first edition, we are pleased to receive Robert Quast from the CBS within the FBI Montpellier node. Robert is a biochemistry researcher specializing in the characterization of membrane protein dynamics. Today, he will present his latest project and show us a multicolor single molecule FRET technique unique in France to study G protein coupled receptor dynamics, and also its application to concrete projects in neuroscience to better understand the mechanism of metabotropic glutamate receptors activation and regulation. And if you have any questions during the presentation, feel free to post them in the chat at any time, and I will read them at the end of the talk when we have a dedicated live session of questions and answers. Robert, the floor is yours.
So yeah, thanks a lot for the invitation, for the introduction. And it's a big pleasure today to present you some of our more recent work, but based on a collaboration that has been going on for quite a while now on using single molecule FRET in 2-color and in 3-color to study membrane protein dynamics on the example of metabotropic glutamate receptors. So, okay, this is not working. Now it's working. So first, let me quickly briefly introduce our team, which is the team of integrative biophysics of membranes in Montpellier at the CBS Center of Structural Biology, where we are 6 permanent researchers at the moment, and our focus is on understanding the biophysics of membranes, so including the organization and remodeling of membranes, mechanical signaling, as well as chemical signaling mediated by ligand-activated receptors, membrane receptors, what I will focus on today. And for this, we gather a large set of different expertise, ranging from cell biology, protein engineering, site-specific protein modification, atomic force microscopy, super-resolution microscopy, as well as single molecule spectroscopy and NMR. And today I will focus on the single molecule part and how we use unnatural amino acid to design conformational sensors to study conformational changes of membrane proteins. So all the work I will show you today is based on a long-lasting collaboration that actually started already before I joined the lab in 2018, and which was initially initiated by Jean-Philippe Pain together with Emmanuel Marja. Fun fact, it was at an airport where they both were stuck and started discussing where they developed this project, and which is now still ongoing and includes now as well Philippe Rondard. So as you can see, it was funded by several joint grants, INR grants, and we've published a few papers over the last years. The last 4 I will focus on today. And all this was all along supported by the FBI infrastructure through a microscope that Emmanuel Marja has developed and which is accessible through the infrastructure. So the biological topic today will be metabotropic glutamate receptors, which are brain receptors, which are G protein coupled receptors. As you may know, G protein coupled receptors or GPCRs are the largest superfamily of membrane proteins encoded in the human genome, with more than 800 members, and they're involved in a variety of different functions. These mGlu receptors are class C GPCRs or glutamate receptor family members because they're somewhat particular compared to other GPCRs. They have a large extracellular domain, as you can see down here, and they form dimers, obligatory for their function and covalent, mediated by a disulfide bridge between the extracellular domains. So these dimeric receptors assemble from 8 different subtypes. And overall, what they do is they sense L-glutamate at the synapse and in other brain cells in order to regulate neuronal excitability and synaptic transmission. What is surprising is that for a long time, they're considered highly interesting potential targets to treat brain diseases. But to date, there's not a single molecule on the market that actually targets them, which emphasizes that we need to better understand how they are activated and regulated in order to precisely modulate them to treat these diseases. So if you look at the timescales, what happens at the synapse, neurotransmitters are released and there are very fast responses happening, initiated by ionotropic receptors, which are ion channels, and quickly followed by G protein activation. G proteins being the effector proteins of the metabotropic glutamate receptors. So the G protein activation happens in the millisecond, tens of milliseconds range, and thus we can conclude that fast conformational changes that have to precede this downstream effector activation regulate synaptic transmission. So what we're interested in is to understand how these receptors transition between states and how this can be modulated by different ligands, for example. So here you see two cryo-EM structures that have been solved within the last 2 years, an inactive resting state and an active state in complex with the G protein, the downstream effector. Here for the metabotropic glutamate receptor 2 homodimer, which I will focus on at the beginning of my talk. So from a structural point of view, the receptors, as I said, are dimers, covalent dimers, which comprise a venous flytrap domain, composed of an upper and a lower lobe, in between which the autosteroid ligands, so glutamate, will bind. This VFT connects to the 7-trans membrane domain, which is a shared feature of all G protein coupled receptors via the cysteine-rich domain. And what we can infer from these static high-resolution snapshots is that large conformational changes occur upon activation. If you compare the structure on the left and on the right, we see a closure of the upper and lower lobes of the venous flytrap domain and a reorientation of the two subunits wrapping around each other and changing the interface of the 7TM domains. However, these snapshots remain static. So, and what you need to get these high resolution snapshots are various stabilizing agents. So what you want to do for high resolution cryo-EM, for example, you want to remove dynamics. This is shown here by the red features like nanobodies, ligands, and point mutations. So what we're interested in is to complement these snapshots in order to understand how receptors transition from one state to the other. Are there intermediates? What's the actual conformational path? So are domain movements correlated or independent? And what are the timescales of interconversion? And for this, we use single molecule first resonance energy transfer, or single molecule FRET, which is a non-radiative energy transfer between a donor and an acceptor fluorophore, here given by the green and the red circles. And we can use this as a spectroscopic ruler to measure the distance between the probes once they have been attached to the protein of interest. We can follow dynamics in real time down to the nanoseconds and up to the minutes time range. And finally, we can do this on single molecules, which we in our lab mainly do using confocal microscopy. So we study freely diffusing molecules, which is an advantage because we do not have to immobilize the molecules to a surface. However, here talking about membrane proteins, what we have to do is we have to extract them from a natural membrane to have them freely diffusing in solution. So all the data I will show you today have been obtained in detergents that we optimize to maintain the functional integrity of these receptors. So in this way, we can get a time resolution of in the sub-millisecond range and up to several milliseconds, which is given by the diffusion time of the receptor through the confocal volume. So before we come back to the biological system, let me just briefly make a point about why single molecule information is highly interesting, even important if we talk about structures. So imagine a static scenario where you want to study your protein of interest, and you assume that it may adopt three different conformations: an inactive red, an intermediate yellow, and a green active. So if you now look at a lot of molecules at the same time and you integrate the information over an ensemble, let's say, you may get the yellow signals. Your conclusion would be that all your receptors, all your molecules are in an intermediate state. If you now start to dilute, you start to resolve red, yellow, and green. So you would change your conclusion. You would conclude that your molecule populates all three states. But now only if you dilute enough and you actually manage to obtain single molecule data, you suddenly may get a completely different picture, which is the red and the green. So your molecule populates two states, and the yellow you saw before was just a matter of averaging over the two different states. The same holds true for a dynamic system. So imagine your molecule populates two states with a fast exchange between the two. So again, if you look at multiple molecules over a given integration time and you average the whole signal, you may again get a yellow signal, which is not true. And now if you look at a single molecule, you may have the same problem, because if your integration time is too long, again, you get the yellow signal. Only if your time resolution is sufficiently high, so higher than the transition time between the two states, you can actually resolve the individual states. So this is why single molecule information is actually important if you want to understand the conformational landscape of a given protein. So here is the setup that we're using, home-built setup, comprising pulsed interleaved excitation and multi-parameter fluorescence detection, in short PIMFD. So just to quickly go through, we use a pulsed white laser source, a super continuum laser, so covering the full visible spectrum. A pulsed laser at 20 megahertz, so every 50 nanoseconds it sends a pulse. We then split the beam in two. One is our donor excitation, the prompt path. And just by placing a filter, we can filter the wavelength we want to excite with. And the second path, we introduce an optical delay to achieve this interleaved excitation or alternating excitation. So every 25 nanoseconds, we will excite either with the green or the red in order to excite the donor or the acceptor fluorophores. And then in the emission, we're separating, so we excite our sample with polarized light, and then we separate by polarization and have two detectors each for each color, the donor and the acceptor. So using a time-correlated single photon counting and APDs, we can then get information on the rastrometric FRET, the excited state lifetimes, fluorescence fluctuations, which can be used to study dynamics, and the anisotropy of the fluorophores, which gives us an idea of the free rotatability. Furthermore, we can use the pulsed interleaved excitation to plot the stoichiometry, which is a ratio between donor and acceptor fluorophores per single molecule, which is very powerful because from the data, we can subtract only double-labeled molecules. And we can get ideas on photophysical artifacts. So overall, this allows us to perform dynamics measurements down to the nanoseconds, up to the milliseconds. And FRET allows to study sub-nanometer distances. Okay, let's come back to the biological systems. So our receptors, I told you, they're dimers. And the orthosteric ligand binding site for glutamate is found in between the upper and lower lobes of the venous flytrap domain. So what glutamate does, it's a full agonist. From a pharmacological point of view, it will give a maximum pharmacological response, while partial agonist that would bind the same site would give a partial pharmacological response. A neutral antagonist will keep the receptor in the basal state. So with regard to drug development, A problem arises from the fact that the glutamate binding site, the orthosteric ligand binding sites, are highly conserved between the different MDR subtypes, which makes it difficult to design subtype selective drugs. Much hope comes from allosteric modulators, which can be either positive or negative, and which bind to the 7 transmembrane domains of these receptors. So what these molecules do is they modulate agonist action either in a positive or negative way, so by increasing or decreasing efficacy and or potency. So why they are interesting is because the binding sites are much less conserved between the different mDOR submembers, and thus there was much hope to develop selective drugs using these sites. So some years ago, we set out to understand how at the structural, at the conformational level, these allosteric modulators modulate the action of agonists over a long-range link of almost 10 nanometers between the VFT and the 7 transmembrane domains. So as I told you, we work on receptors extracted from natural membranes, so in detergent mice cells in a confocal setup. So the first step was to actually find conditions to extract the receptors while maintaining their native ligand responsiveness, but also to have them be functional over extended measurement times, because our measurements typically take a few hours to overnight. So here you see a set of different detergents that had previously been used for metabotropic glutamate receptors and which we screened for our purpose. And the functionality we basically measured using a FRET sensor here at the ensemble level, not yet single molecule. This will come later. So by activating these FRET sensors, these receptors with glutamate, we start from a high FRET. Here on the right, you see the curve, a dose response curve. We start from a high FRET in the inactive, and then upon increasing concentrations of glutamate, the FRET decreases, the receptors reorient, and we measure the activation. And then using these allosteric modulators in combination with the glutamate titration, we can get an idea of the functional link between the 7TM domain and the VFT domain, given by the shift of the blue curve here compared to the black curve to the left. So an decrease in the EC50 value and an increase in the Emax, the efficacy. What we can then do is we do this at different time points after solubilizing the receptors from membranes, and we can get an idea of the functional integrity over time by calculating this delta EC50, for example. This is what we did for all the different detergents, and as you can see, most detergents are not suited to maintain the receptors in the functional form over up to 24 hours. So on the x-axis, you have time 0, and on the y-axis, time t24 hours. Only the mix of LMNG, CHS, GDN gave a response that was the same at T0 and T24, similar to our control of receptors in crude membrane fractions. So to make another point here, as you can see, two detergents that didn't work well, they don't show a PAM effect, but what you can see is that they show a nice dose response curve to glutamate. So essentially, we see a FRET change, a response to glutamate, but the functional link between the 7 transmembrane domain and the VFT is not intact. So take-home message, you need proper functional assays before you do single molecule FRET. Otherwise, you may see conformational changes in dynamics, but they may not be transferable to what happens in the native environment. So with this, we performed our first single molecule FRET study on the metabotropic glutamate receptor 2 homodimer and its allosteric modulation. For the sake of time, I will not go into details today. So the first study, we used SNAP tags attached to the N-terminus of the two subunits. But since then, we've mainly changed our labeling strategy because these SNAP tags, they're fairly big compared to the size of the receptor. So you see 19 kilodalton compared to 190 kilodalton of the dimer. If you attach 2, you're already at about 20% of the molecular weight of the native receptor. So what we're now routinely using are non-canonical amino acids or unnatural amino acids, if you like, that we couple to fluorophores to introduce small chemical handles and reduce the impact of labeling. So overall, this way we can place these unnatural amino acids all throughout the structure where we want. So we can go beyond N-terminal labeling. We can minimize the invasiveness of the labeling. And as we showed some years ago, by using orthogonal compatible chemistries, we can even improve the accuracy of single molecule thread measurements. So how can we introduce these unnatural amino acids into the protein where we want them? So you all know ribosomal protein synthesis, I assume, and the genetic code with 64 codons. 61 are sense codons coding for amino acids, and we have 3 stop codons. So what we can do is we can choose one of the stop codon. We will introduce it into the open reading frame of our gene to be expressed at the position where we would like to label. And then by introducing into the system an orthogonal tRNA, amino acid tRNA synthetase pair together with a suppressor tRNA with an anticodon complementary to the stop codon we would like to reassign, and then adding an unnatural amino acid to the medium, we can actually achieve, if this system is orthogonal, which means it doesn't cross-react with natural amino acid tRNA synthetase pairs, we can achieve a reassignment of a stop codon to make it a sense codon for our unnatural amino acid, which will then be incorporated at the position we would like. In this way, we can incorporate chemical handles, like here, an example that we developed a few years ago with our collaborators at the IGF, a propagyltyrosine or propagylloxyphenylalanine bearing an alkyne group, which we can couple to azide functionalized dyes, here picolyl azides, via copper click chemistry. So in this way, we set up a set of different sensors, and at first, let's look at 2 of them. So one is the monitoring the closure of the upper and lower lobes of the venous flytrap, the VFT domain, upon ligand binding, and the other one on the reorientation of the upper lobes, similar to what I showed you by the ensemble thread before for the functional assay. So here's the data on the left, the reorientation, on the right, the closure. So in the absence of ligand for the reorientation, we find a major high thread population corresponding to the fluorophores being fairly close. If we now add a partial agonist, DCG4, you see a low thread population appears, but at the same time, we still see the high thread population. So from this, we can conclude that this partial agonist establishes a dynamic equilibrium between resting, inactive, and active conformations. Why do we know it's a dynamic equilibrium? I told you before that our receptors spend roughly 3 to 5 milliseconds on average in the confocal volume. So the fact that we detect 2 populations means that the transition between these 2 states is slower than the observation time of our approach. If we now add the full agonist glutamate, we see a similar picture, but with more molecules populating the active low thread state. We can now add our positive allosteric modulator, BINA, and we see a strong shift stabilizing the active state. The same happens if we mix the PAM with glutamate. We can then add an antagonist, and we see it doesn't change. It keeps the receptor in the basal resting state. If we now compare the thread efficiency we determine, you can see that they're highly similar for the resting and the active state. So from this, we can conclude that partial agonists, for example, that give a partial pharmacological response, likely at least at the resolution of our sensor, do not do this by stabilizing intermediate signaling active states, but rather by just playing on the equilibrium between active and resting. Okay, so for the VFT closure, what we expect in the absence of ligands, we get a low thread, so the VFTs are open, the lower and upper and lower lobes are well separated. If we now add a partial agonist, again, we get a 2-state equilibrium between open and closed. We can then add glutamate, the full agonist, which gives an almost complete closure, so shifts the equilibrium strongly to the right. If we now add our PAM with the partial agonist, again, we see a small effect stabilizing the closed state. And now, interestingly, when we mix glutamate with the PAM, we don't see any further effect as compared to glutamate alone. If we now add our antagonist, we see a slight closure, but not corresponding to a fully closed VFT domain. So what we found very surprising here, again, and if we compare the thread efficiencies, you see that the active state, the closed state, seems to be highly similar between all the different conditions, while the low thread state is somewhat ligand dependent. So what we found highly interesting is that the reorientation is incomplete. So there's a dynamic equilibrium when activated by the full agonist glutamate, while the VFTs are fully closed. We then quantified this here in biological replicates. On the left, again, the reorientation, on the right, the closure. And you can see in blue with the PAM and in black only the agonist, you see a clear shift towards the active state. And for the closure, we barely see any shift for full agonists when we add the PAM. We also did this for the lower lobe sensor and the CRD sensor, which give a similar picture as the upper lobe sensor. So glutamate establishes a dynamic equilibrium and the PAM shifts almost fully to the active state. So from this, we came up with a model. We figured that if there are resting and inactive populations, but all VFTs are closed, there must be an intermediate state. So our first model was based on the idea that agonist establish a three-state equilibrium between the resting open-open and the resting closed-closed and the active closed-closed. And pumps act on the second equilibrium to stabilize the active closed-closed state. This is nice, but it's somewhat indirect to conclude from two individual two-color sensors that there must be this intermediate state. So we decided to go a step further and introduce a third fluorophore in order to monitor the reorientation and the VFT closure for each single molecule simultaneously. So this is the work of Leo Bonhomme, a talented PhD student that defended beginning of the year. And he set up a site-specific triple labeling strategy because at the time we wanted to do this, there was no strategy available using mammalian hex cell expression for what we wanted to do. So he constructed two constructs of the mGlu-R protomers, one with an N-terminal snap tag and a TAG stop codon for unnatural amino acid incorporation, and the second protomer without a snap tag and with a TAA to incorporate a second unnatural amino acid. So what he did is in hex cells, he expressed the system and incorporated in parallel two different unnatural amino acids in response to two different stop codons, which are shown here. So the PRF or propagyl tyrosine or phenylalanine, which I showed you before, and the a transcyclooctene TCOK. And then perform triple labeling. First, the snap tag on the upper lobe of one protomer, then the TCOK with the tetrazine dye, and the corporate click chemistry of the propagyl oxyphenylalanine with picolyl azide to achieve this triple labeled molecule. And then he tested whether the incorporation is specific. So PRF is only incorporated in response to the TAG codon and TCO in response to the TAA codon, which is important because, I'm way over time. Okay. He also verified that the labeling reactions are specific to really get only the desired product, and then measured single molecule FRET with a special focus on the VFT closure, so FRET between donor 1 and acceptor 1 and donor 2 and acceptor 1. So we then teamed up with the lab of Don Lamp in Munich, where we performed the three-color single molecule FRET measurements. So from these measurements, what we can do is we can do a 2D, we can construct a 2D plot of the VFT closure here shown in, on the x-axis and the reorientation on the y-axis. So for our resting open state, we would expect it to be here at the lower left bottom, while the active closed should be at the upper right. And our resting closed should be found at the lower right bottom. He then performed the measurements here in the apo condition, and we found what we were expecting, a resting closed state at low FRET, low FRET. And then interestingly, when he added glutamate at a saturating concentration, we were able to recover the active closed state here on the top right. However, the bottom population, so low green-red FRET, was neither corresponding to the resting open, nor the resting closed state, but was found in between these two. We then added another layer of analysis together with Don Lamp, a three-color photon distribution analysis, which is essentially fitting the data to different models of different states. And what we recovered, when you look only here on the right, for the sake of time, I'm speeding up a little bit, where we transformed into distances. You see the resting open at a high distance between the upper and lower lobes and the two protomers. And then at the bottom for the glutamate, you can see that our intermediate conformation is actually intermediate between the open and the closed. So from this, we concluded that we found a new intermediate state and that our conclusion before of the resting closed state was actually wrong. So we corrected our model and replaced the intermediate state by a resting semi-closed state. So to sum up the first part, I hope I convinced you that agonists induce dynamic equilibria. In the case of these metabotropic glutamate receptors, partial agonists are less potent to populate the active state rather than stabilizing intermediate signaling active states. Allosteric modulators stabilize the fully active state, and we think that we describe a new novel agonist induced intermediate state that has not been seen in cryo-EM, likely due to dynamics. And now, how much time do I have? A few minutes? Okay. I think I will have to skip the second part. Well, I will make it quick. So I told you that the glutamate receptor can assemble in heterodimer in homodimers from 8 different subtypes, but they can also assemble in heterodimers, which has been shown years ago by heterologous expression and now has been verified in the brain of rodents, for instance, for the 2,4 heterodimer. And even more interestingly, the 4 subunit, when expressed in the same cells as the 2 subunit, prefers heterodimerization. So this gives a whole new idea of synaptic regulation through homo and heterodimers. And while we're not yet understanding their physiological function, these are interesting new targets for drugs to modulate neural excitability and synaptic transmission. So together with collaborators in China in Wuhan and the IGF, they performed pharmacological measurements and produced cryo-EM structures to get an inactive structure of the mGlu24 heterodimer and a fully active structure in complex with G protein, as well as 4 intermediates when activating one or the other subunits with subtype-specific ligands. And we perform single molecule FRET measurements to better understand the conformational equilibrium of these intermediate states. So in the APO, you see, again, an upper lobe sensor like the one I showed you before, a high FRET state. If we now activate the mGlu4 subunit specifically, we see the appearance of a low FRET state. So again, a dynamic equilibrium between resting and active. If we activate the mGlu2 subunit, we get a similar picture. And now, very interestingly, if we activate with glutamate, which binds both subunits, we get a full shift, we get a full stabilization of the active state, in contrast to what we saw for the mGlu2 homodimer. We then also included G protein, the downstream effector, and you see in the apo state, it's highly similar to the apo state without G protein. And now if we activate one subunit and we add the G protein, we see it acts like an allosteric modulator, shifting the equilibrium towards the active for both subunits. So to sum up, glutamate efficiently stabilizes the active orientation, single ligand dimer sample 2 states, and GI acts as a palm. And with this, let me finish. And just point out that here I only showed you a long-lasting collaboration on metabotropic glutamate receptors, but obviously these techniques can be applied to other systems. So some of our ongoing collaborations through the FBI infrastructure include other G protein coupled receptors to study activation, allosteric modulation, and signaling selectivity in response to modulatory proteins, pathological mutations, or the lipid environment, for example. We've also an ongoing collaboration on ion channels, as well as how bacterial toxins form pores. And finally, as a teaser, so our home belt setup will be updated to 3-color beginning of next year, which will also be available through the FBI infrastructure. And we just purchased a new microscope, a commercial single molecule FRET microscope, which we will combine with AFM, which will allow to do correlative confocal imaging and AFM, as well as, for instance, simultaneous single molecule FRET measurements and single molecule force spectroscopy. And with this, I'm going to stop. Just to quickly thank my team members, Emmanuel Marja, Leo, and Caroline, who were directly involved in the project, our collaborators at IGF, Perkinelmer Bio, now called Revity, the University of Leipzig, Irene Coyne, Jan Feng from Wuhan, Don Lamp, where we did the 3-color measurements, which we will now establish in France, and all the funding partners and France-BioImaging as well, and you for your attention, and I'm open to answer your questions.
Oh, thank you very much, Robert, for your insightful presentation. So it's time for questions and answers. So if you have any questions, please raise your hand and I will let you talk. Oh, we have a question in the chat. So it's Mohammed, and it tells us, thank you for showing us this beautiful research. I am a student and do not have good understanding of single molecule biophysics. My question is, what is stopping us from studying these dynamic structural changes with a single molecule of proteins instead of population of molecules?
I'm not sure I understand the question, or I did not at all manage to make the point in the beginning when I talked about single molecule. So I will try to answer, but I'm not sure if I understand the question right. So the problem of a population of molecules is that if you integrate the signal, your readout from this population, you just get an average. Cryo-EM, we talk about single particle picking, but in the end, you average over thousands and thousands of molecules. So you will get an average result. So you will not know, as I showed before with the 3 colors, green, red, and yellow, if, for instance, all your molecules are in an intermediate state, yellow, or actually just green and red, and the average would be yellow. I don't know if I answered
Mohammed, it is okay for you, or do you want to talk maybe?
Well, maybe we can continue, and Mohammed,
Yes, yes,
if he
yes.
wants, he.
We have a question from Alexei. Do the detergents form micelles in solution, and is their curvature an issue to model membrane receptor structure? Sorry for my accent.
Definitely. So detergents form micelles. This is actually wanted to keep the receptors in solution, freely diffusing. You want to stay above the critical micelle concentration. And the curvature is likely not the curvature we see in cells. So, and this is difficult to mimic in detergents. So I'm completely agree with you that other membrane biomimetics or even going to living cells is what we should aim in the future.
A question from
Hmm.
Morgan. What are the barriers to moving to living cells?
Okay, good question. So the first challenge, so let me just point out that it has been done in living cells with the SnapTech labeling strategy for membrane proteins. There's a single example to my knowledge. Now there are a few now, but so these SnapTechs, they're big. You can attach them to the N-terminus. This is super nice, but you will essentially just measure a single conformational change. So you cannot place the fluorophore on the protein where you wanted to study different conformational changes, different domain conformational changes. So what we're trying to push is using this unnatural amino acid strategy to label proteins and do single molecule FRET in the membrane of living cells. But what's challenging is to achieve specific labeling in the membrane of living cells, because if you reassign a stop codon to incorporate your unnatural amino acid and label, then the cell uses this stop codon to terminate the translation of proteins. And so you will find your unnatural amino acid in endogenous proteins to a low extent, but this happens. And thus you will label other proteins than your receptor of interest. So currently this is what's limiting. And the second limiting step that is somewhat challenging is achieving very low expression levels, but at the same time, a very high amount of labeling efficiency, because you need two fluorophores on the same molecule, but then you need a low concentration of labeled molecules to be able to detect single molecules. Otherwise, you average again. So these are, I think, the main two limitations at the moment, but I think it will not be long until we can do this.
Alexei, would it be possible to use flat artificial membranes?
Sure. There are examples in the literature where people have used nanodiscs, for example, which are like flat membranes surrounded by a scaffold protein. You could imagine supported lipid bilayers. Yeah, why not? You could even use liposomes. The thing is that with liposomes, you would need to ensure that you get one single molecule per liposome. Feasible, maybe not so easy.
Okay,
Ah, okay.
Mohammed, it's
Mohammed.
okay.
Yeah, sorry. Mhm. Go ahead.
So the question from Mohammed, I was thinking why the last diagram had a lot of data points. Isn't a seeing change in one single molecule allow us to draw a conclusion for all the molecules?
Okay, I don't know. Maybe you mean something like that? Yeah, maybe I didn't properly explain how we plot the data. So on the y-axis, you have the occurrence. This is the number of molecules. So for each single molecule, we determine the Fred efficiency, which is the x-axis. And then we plot them, and then you get these distributions. So in order to get this distribution, we need to measure many single molecules, because one single molecule will spend 3 to 5 milliseconds in the confocal volume, and we will get maybe 100 photons or something. So in order to actually draw conclusions, we need to measure many molecules. So here normally we measure around I don't know, 2,000 molecules up to 10,000 if we want to go even deeper in the analysis.
I have a question.
Yes.
Robert, can you come back to the board with all the subgroups of mGlu of mGlu receptor? Um, yes, this one. Um, um, do we know all the other subgroups mechanism or not yet?
Um, Good question. So you mean from a structural point of view?
Yes.
So we have at least one structure for each subtype, except for six, I think. Six is special because it's in the retina, so it's a bit different than all the others. We have a single structure at least, but I'm pretty sure we do not have active and inactive structures for all of them. And with regard to the intermediate states, I think we do not know much, except for mGlu5, which has been studied quite a lot now, and mGlu2.
Okay. And uh, we know that these receptors are associated to neurological disorders, but do we know how it works?
Sorry, what? How what works?
Um, 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 do we know? Um, sorry. Do we know how this receptor uh, act in these neurological disorders? What is
Okay.
the link between the receptor and
Yeah.
the disorder?
Yeah. So I'm not an expert in this. But what I can tell you is that, as far as I understand, these receptors are not the cause of the diseases. But as they're so central to synaptic, to neuronal excitability and synaptic transmission, they may be targets to treat these diseases.
Okay,
And then, well, I guess the idea that we have which receptor is involved in which disease comes from animal models, from pharmacological studies. But to my knowledge, we do not well understand yet the exact expression patterns of the different subtypes in the different neuronal cells. Usually what we know is in a brain region, these receptors are more abundant. But this doesn't mean that in the same cells, for instance, mGlu2 and mGlu3 are expressed in the same cell or not. So I think the whole idea of heterodimers um, may bring us closer to understanding how the different subtypes, the different dimers regulate these mechanisms and how they may be implicated in diseases.
okay, thank you.
Okay.
Uh, we have questions from Pavel. Um, how do you define the threat efficiency for the first question? And the second question is labeled receptor biologically functional and can transduce a signal?
Okay, good question. So the threat efficiency, all the data I showed you are basically fairly simple. Um, um, they're ratiometric threat. So essentially, you excite the donor, you look how much signal you have in the acceptor channel, right? Um, at the same time, you can get the threat efficiency from the fluorescent lifetime data, which we also have in the same set of data. So you can look at the reduction of the lifetime of the donor, which is a means to compare to the donor only, where there's no acceptor, to calculate the threat efficiency. The second question, and most of the data I showed you is what we call accurate threats. So we correct for direct excitation, crosstalk of the donor into the acceptor channel, as well as beta and gamma, which are related to, so gamma especially, which is related to the excitation intensities of the lasers and the detection efficiencies on the detectors. Question 2 is labeled receptor biologically functional and can transduce signal. Well, what we showed is that the allosteric modulator has an effect in a way, let's say, well, similar to what we see in our membrane fractions at the ensemble level. We also did dose response curves and we recover the same affinities, so same EC50 approximately from our single molecule threat measurements. And can it transduce a signal? I mean, we tested the effect of G protein on the conformations of the receptor, and this works. So I would say yes, but um, well, maybe you would want other controls to prove that it can actually signal, because what we have not measured is the G protein activated and then signals. Yeah, I cannot tell you yet. So I think we're as close as we can get for the moment.
Okay. And maybe the last question, because we have 7 minutes left. So a question from Morgan. So if I understand correctly, the graphs shown to answer Mohammad's question, each each occurrence is a snapshot of a single molecule in a given state as it passes the focal volume. Will we ever see the dynamics of conformational changes in single molecule mode? Could the molecule stay long enough to move from resting to active while imaging it?
Yeah, so that's a good point again. So here what you see is individual states. So the dynamics are slower than our observation time using confocal microscopy on freely diffusing molecules. So what we can do is we can immobilize these molecules and look at them by turf microscopy, where you can monitor longer conformational changes. So you essentially, with a camera-based detection, look at the same molecule as long as your fluorophores stay intact, so until photobleaching. So you can reach seconds to minutes in measurements like that. But the response time of the camera, so the detection, the response time, the time resolution of a camera-based detection is in the tens to hundreds of milliseconds. So in the confocal approach, we look at the molecules for, let's say, 3 to 5 milliseconds on average. And with TURF, we could look, let's say, at the maximum, maybe with a resolution of 10 milliseconds. So there's still this gap between, let's say, 3 and 10 milliseconds, right? So what we can do now and what we're planning to do once we have the new Luminosa microscope, we want to, and there we would go to nanodiscs. We want to immobilize the receptors and then do the confocal approach on them. So we will not lose time resolution, but we can look at the molecules for potentially up to seconds or even minutes. And in this way, we should be able to actually see the dynamic transitions and quantify them. Now, if you have dynamics that are slower than the observation time here in the confocal approach, there's a set of different methods how to identify so fast conformational changes below the millisecond range. So the microsecond and nanosecond range, there are different approaches how you can basically dissect the data and obtain information on these fast dynamics. So you can have a look in the publication from us 2021, where we did this a little bit on the SNAP labeled receptors. And could the molecule move first? Yeah, I hope I answered the question.
Okay. Thank you, Morgan. Thank you, Pavel. Thank you, Mohamed, and everyone else for your questions. If you have any further questions, feel free to contact us or maybe directly Robert. Our contacts are available now in the chat and we also appear in the replay. Thank you, Robert, for being the first to present your research. And thank you, everyone who joined us today for FBI Connect. And for your information, the presentation has been recorded and will be available on YouTube if you want to watch it again or share it with your colleagues. Goodbye, everybody. Have a nice day.